Welcome back to the Deep Dive. Today, we're jumping right into something pretty amazing, a real technological revolution happening at the microscopic level. 3D organoids.
Yeah, these are fascinating.
These human stem cell-derived mini-organs, well, they're promising to totally change how we remedy diseases, really providing that link we desperately need between basic science and, actual human biology.
It's a huge leap, absolutely essential. I mean, for decades, researchers basically had two main tools. Cells grown flat on plastic dishes.
Right, 2D cultures.
Exactly, 2D. Or animal models, usually mice or rats. And let's be honest, for a lot of complex human diseases, those models, they've kind of hit their limits.
We got the problem with 2D cultures. They're too simple, no real structure. But animal models, they're whole living systems. Why are they failing us? Where's the gap?
Well, it often comes down to poor concordance. Basically, they don't match up well enough with human biology. Think about heart function, for example, or how our human liver processes drugs. That's a big one. Or even our immune system. Rodents just, well, they don't work the same way we do. So the challenge is, how do we build a really accurate, human-only model in a dish that actually captures all that complexity?
And that's our mission for this deep dive. Our sources are pointing to two really critical missing pieces that are holding back these otherwise brilliant mini-organs. First, they usually don't have a working blood supply.
The vasculature.
And second, they often lack key immune cells. So we need to dig into how these next-gen platforms are tackling this using some really tough conditions like traumatic brain injury and inflammatory bowel disease as examples.
Exactly. And it's worth saying what organoids already do is pretty incredible. They're like feats of biological engineering because they self-organize.
Right.
You just give the stem cells the right signals, the right environment, and boom, they start forming structures that look a lot like, say, the cerebral cortex in the brain, or that complex lining of the intestine, the cryptovillus structure.
And they actually work functionally too, right? We see neurons firing spontaneously, forming circuits in these mini brains. But then there's this physical problem when we try to, grow them up. Why can't they just get bigger?
That's the core physiological barrier. It's called the diffusion limit.
Diffusion limit, okay.
Yeah, think about it this way. In healthy tissue in your body, almost every cell is super close to a tiny blood vessel, a capillary. Usually no more than maybe 100 to 200 micrometers away. That's tiny, a fraction of a millimeter.
Okay, why that specific distance?
Because that's about the maximum distance oxygen and nutrients can effectively travel just by passive diffusion, just soaking through the tissue.
So if the organoid gets bigger than that, the stuff in the middle is basically cut off. It starves.
Precisely. So if a brain organoid grows to, let's say, 1 or 2 millimeters across, which isn't very big, it develops this necrotic core.
A dead zone.
A dead zone right in the center. And that obviously messes up long-term growth, keeps the organoid kind of stuck in an early developmental stage, like embryonic or fetal. So if we want to study diseases that affect adults, we need vascularization, a built-in blood supply. It's really the only way to get the size and lifespan needed for them to mature properly.
Okay, so we've got the infrastructure problem, the vessels. Now let's talk about the security system, the immune part. What's the most critical immune cell that's often missing in these brain models?
That would definitely be microglia. These are the brain's own resident immune cells, and they are absolutely vital. They do general housekeeping, maintaining neural homeostasis, but they also do something really critical called synaptic pruning.
Synaptic pruning. Sounds like gardening almost.
Yeah.
What's it actually doing in the brain?
It's actually a great analogy. It is like gardening for your brain circuits. Microglia are constantly moving around, especially during development, but throughout life too. And they actively trim away synaptic connections that aren't needed or maybe damaged.
Cleaning things up.
Exactly. It's crucial for efficient learning, for memory, basically for keeping the neural network lean and effective.
So if the microglia are missing, the model can't really react properly to something like trauma, right?
Precisely. They're the first responders in the brain, so not having them is a major limitation if you're trying to model anything involving inflammation or injury, like TBI, traumatic brain injury. Researchers are trying a couple of main ways to get them in there.
Okay, what are the strategies?
Well, the first one is basically co-culture. It's pretty direct. They just take microglia or sometimes stem cells that are programmed to become microglia and mix them right into the developing organoid, kind of hoping they'll integrate.
Just add them in.
Right. The second method is a bit more sophisticated. It's called endogenous development.
Okay.
Here they use tools like genetic engineering or specific signaling proteins called cytokines to actually encourage the stem cells already inside the organoid to turn into microglia themselves.
Huh. Why go through all that trouble with endogenous development if you could just add the cells?
Yeah.
Seems more complicated.
It is more complicated, but the idea is that a cell that develops within the system endogenously is likely to be better integrated.
Ah, more natural.
Yeah, both physically and functionally, it should lead to a more physiologically relevant model where the microarrays really belong there, maybe communicating better with the neurons around them.
Got it. Okay, let's circle back to the infrastructure of the blood vessels.
Right.
Building those, it sounds like it's about more than just delivering food and oxygen, right? What else does that vascular network do?
Oh, absolutely. It's like a whole system upgrade. It's not just nutrient delivery. The vasculature allows for really important communication crosstalk between the main cells of the organ, the epithelial cells, and the vascular cells themselves.
Okay.
And maybe most importantly, the blood vessels are the highway system. They transport signaling molecules, hormones, immune cells between different parts of the body, between different organ systems. That's critical if you want to model systemic diseases.
Makes sense. So how do you actually build such a complex, tiny network of vessels in the lab? What are the options?
Well, there are sort of two main philosophies. The first is very engineering driven. It's called vascular patterning.
Okay.
This uses fancy fabrication techniques like 3D printing or something called soft lithography to create these predefined hollow tubes, microconduits. Then they coat these tubes with endothelial cells, the cells that line blood vessels. Essentially, you're building the road network first.
Sounds very precise. What's the downside?
Well, the geometry often isn't quite right. You end up with channels that might be square or rectangular in cross-section.
Not like real blood vessels.
Not really. And that shape actually affects how the fluid flows and the sheer stress the endothelial cells feel, which is important for them to behave naturally. Plus, these patterned vessels aren't very dynamic. They can't really sprout new branches or remodel themselves like real vessels do.
Okay, so if the top-down engineering approach has limits, you let biology do the work, what's the other method?
That's self-assembly. And this is more bottom-up, biology-directed. Researchers take the endothelial cells, mix them with some supporting cells called stromal cells, and put them all randomly into a soft gel, like collagen or fibrin.
And then what happens?
Then you just wait. Over a few days, the cells start moving, they organize themselves, line up, form hollow tubes, lumens, and spontaneously create a whole interconnected network of capillaries.
Wow.
Yeah, it usually results in vessels about 15 to 50 micrometers wide, which is very similar to what you see in the body. It mimics natural processes like vasculogenesis and angiogenesis.
Right, those terms. Vasculogenesis and angiogenesis. What's the difference again, and why is self-assembly tapping into both?
Good question. So vasculogenesis is basically building vessels from scratch, like in early development. Angiogenesis is when new vessels sprout off from existing ones, like during growth or wound healing. The cool thing about self-assembly is that the cells naturally do both. They build the initial network, and then they can sprout and refine it. It leads to a much more organic, integrated, and dynamic network compared to just building fixed channels.
And you mentioned flow earlier. How does that play a role here?
Flow is key. Researchers often engineer microfluidic devices that create a very slow, controlled flow of fluid through the gel itself. That's called interstitial flow.
Okay.
This flow does more than just deliver nutrients. It actually provides cues that guide the newly forming vessels to connect up properly to anastomose with adjacent microfluidic channels that act like larger vessels. This makes the whole network perfusable. You can actually flow liquid through it.
That is some seriously impressive biological engineering. Okay, let's shift to how this is being used. How are these advanced mini tissues helping us understand specific diseases, starting with traumatic brain injury, TBI?
Yeah, a really important area is mild TBI or MBTBI, especially the kind that comes from repeated low-level blast exposures, or LLB.
Right, you hear about this with soldiers or law enforcement?
Exactly. We're talking about those subconcussive blasts, often less than 4 CI, maybe from using heavy weapons repeatedly. They don't cause obvious immediate damage, but the cumulative effect over time can be devastating, and it's been really hard to study mechanistically.
And these organoid systems seem perfect because you can precisely control the force, the biomechanics, without the complexity of a whole animal.
Precisely. The setup usually involves specialized loading devices, things that can deliver a controlled blast wave or impact right onto the organoid in the dish. Then they combine this with the advanced organoids we've been talking about and use non-invasive ways to see what's happening over time.
And what are they seeing? Are the organoids showing TBI-like damage?
They are, yeah. Studies have managed to replicate key TBI indicators. They see things like an increase in reactive oxygen species, that's oxidative stress, and leaky cell membranes after a simulated blast. They also see increased cell death, neuronal damage, and using techniques like RNA sequencing, they can actually map out the specific inflammation and cell death pathways that get activated.
Okay, but to track these changes over time, especially subtle ones, without damaging the organoid each time you look, you need some pretty clever imaging techniques, right? What label-free methods are they using?
Right, non-invasive is crucial for seeing how damage evolves. One key technique is full-field optical coherence tomography, or FFOCT.
FFOCT. What does that show you?
It's great for looking at structure in 3D, almost like microscopic ultrasound, but using light. It lets researchers see changes in the internal structure of cells and tissues over days or even weeks without adding any labels.
Structure is one thing, but what about function? Like how the cells are actually working or struggling metabolically?
For that, they often turn to fluorescence lifetime imaging microscopy, or FLIM.
Okay.
This technique looks at the natural fluorescence of molecules already inside the cell endogenous fluorophores that are involved in metabolism. By measuring how long that fluorescence lasts, which changes based on the metabolic state, FLIM can tell you how hard the cell's energy-producing machinery is working or if certain pathways are failing. It's like looking at the cell's engine.
Very cool. And what about those bigger organoids, the ones getting close to that 2 millimeter size? You mentioned light having trouble getting deep. How do they see inside those?
That's where 3-photon microscopy, or 3PM, comes in.
3 photon.
Yeah. This is a more advanced technique that uses lower energy photons in pulses, which allows light to penetrate much deeper into scattering tissue. It's been a breakthrough for imaging larger samples, letting researchers see inside cerebral organoids up to maybe 2 millimeters deep.
And why is getting that deep view, seeing the core, so important for something like TBI? What would you miss if you only saw the surface?
You could miss the most important part of the story. Think about a blast wave hitting the organoid. The cells on the surface experience the initial impact very differently than the cells buried deep inside.
Right, the forces would be different.
Exactly. The core might experience more compression or shear forces or rebound effects. The cellular response, the damage, could be completely different in the core versus the periphery. If you can't see what's happening deep inside, you really don't have a complete picture of the TBI pathology. You might draw the wrong conclusions.
Okay, that makes total sense. Let's switch gears now from the brain to the gut. Inflammatory bowel disease, IBD, things like Crohn's and ulcerative colitis.
Yeah.
How are intestinal organoids making a difference there?
This is another area where organoids, especially patient-derived organoids or PDOs, are having a huge impact.
PDOs meaning made from a specific patient's cells.
Correct. And that's key because IBD is notoriously heterogeneous. Two people with Crohn's disease might have very different symptoms and respond differently to treatments. Using PDOs lets researchers model the disease as it appears in that specific person.
So you can see their individual biology in the dish.
Exactly. They can look for specific problems like defects in tight junction proteins. Those are the seals that keep the gut barrier intact. And crucially, they can test how that patient's organoids respond to different drugs, like vedolizumab, for example, before even giving it to the patient. It's much more personalized.
And the gut is full of bacteria, the microbiome. How do they factor that in? You can't ignore the microbes?
Absolutely not. So the newer platforms are integrating organoid microbe co-cultures. They're also using these advanced gut-on-a-chip systems.
Gut on a chip.
Yeah, these are microfluidic devices designed to mimic the physical environment of the gut more closely. They can simulate peristalsis, the squeezing motion of the intestines, by applying fluid flow and shear stress. This dynamic environment is really important for studying how an unhealthy microbiome, what we call dysbiotic microbiota, actually disrupts that gut barrier.
That's getting incredibly close to the real thing. I also read they're using CRISPR gene editing with these.
That's right. CRISPR is being used to understand the genetic side of IBD. Researchers can take organoids and specifically knock out genes known to be linked to IBD risk, like the NOD2 gene.
Oh.
By doing that, they can directly connect a specific genetic mutation to a specific problem at the cellular level. For example, they've linked NOD2 mutations to problems with the differentiation of certain immune cells in the gut called Paneth cells. It helps untangle the cause and effect.
Fascinating. Okay, one more big area, cancer. We talked about adding vasculature. When you create these vascularized microtumors, or VMTs, what's the key advantage over the older, simpler tumor models?
It's a massive difference. These VMTs model the tumor microenvironment much more realistically. You can actually watch the primary tumor grow, see how it recruits its own blood supply, that's angiogenesis, and potentially even model metastasis, how cancer spreads.
And our sources highlighted something really compelling about drug testing of these VMTs compared to animal models. What did
Wow.
those studies find?
Yeah, this is really significant. They found that the way cancer drugs worked or didn't work in the vascularized microtumors often matched up very closely with what happened when the same drugs were tested in mice in vivo, which
Wow.
is huge because traditional 2D cultures and even simple 3D tumor spheroids without vasculature often do a terrible job of predicting how a drug will actually perform in a living system, let alone a human patient.
So does this mean VMTs could potentially start replacing some animal testing in preclinical drug screening?
Yeah.
Saving time, money, maybe getting better drugs faster?
That's absolutely the hope, and the potential is enormous. If these VMTs can more reliably predict which drug candidates are likely to fail before they go into expensive, lengthy animal trials or even human trials, well, it could radically streamline the whole drug development pipeline.
Yeah, letting researchers focus only on the most promising therapies.
Exactly. Accelerating the path to finding effective treatments.
It really feels like a convergence point.
Right.
You've got the stem cell biology making the tissue, the microfluidics creating the environment and the flow, and then these advanced imaging techniques letting us see and confirm what's happening.
It really is a convergence, and it's transforming organoids from just being interesting 3D blobs of cells into genuinely high-fidelity, physiologically relevant systems. We've seen how they're tackling those immediate bottlenecks, getting past the diffusion limit with vascularization, adding the immune components.
So where does this lead next? We've got these sophisticated single mini organs, but our bodies aren't just one organ, they're interconnected systems.
That really is the multi-million-dollar question, isn't it? The clear next frontier, the big push now, is developing multi-organ systems.
Linking them together.
Precisely. Physically connecting vascularized organoids representing different organs. Imagine linking a mini liver, a mini kidney, and a mini intestine using a shared circulating vascular network.
Is this the human on a chip concept finally becoming real?
This is exactly where it's heading. And this integration, it's not just cool, it's essential if we want to model diseases that affect the whole body, like cancer spreading, metastasis, or widespread inflammation. And for drug development, it's crucial for modeling ADME.
ADM. Right, that's absorption, distribution, metabolism, excretion. Why can't a single organoid model that?
Because ADME describes the entire journey of a drug through the body. It gets absorbed, maybe in the intestine, distributed through the blood, metabolized often by the liver, and then finally excreted, maybe by the kidneys.
The whole pathway.
Right. If you test a drug only on a liver organoid, it might look fine. But maybe the liver produces a byproduct, a metabolite, that's actually toxic to the kidney. You'd completely miss that toxicity unless you have both organs linked in the system. Coupling these vascularized mini organs lets us finally see that full systemic picture before a drug ever gets near a person.
We've watched researchers chip away at these huge problems, diffusion, immune cells, imaging depth. Now linking systems together. That feels like the ultimate challenge.
It is immense, and maybe the provocative thought to leave you with is this: building the multi-organ system.
Yeah.
Maybe the hardest part isn't actually making the individual mini organs work well. We're getting pretty good at that.
Okay.
The real challenge, the thing keeping researchers up at night, is creating a single universal blood, a perfusion medium that keeps all the different organoids happy simultaneously. Think about it. The liver has very different metabolic needs than the brain. Neurons are incredibly sensitive.
Right. Different needs, different environment.
Exactly. Finding that perfect balance, that physiological Goldilocks zone in the circulating medium that supports a liver, a kidney, and a brain, all dynamically linked, while ensuring the whole system behaves realistically, that is the next massive hurdle. That's the integration challenge we still need to solve.